Hey guys, welcome to StudyIQ. I am your friend Rahul Saigaonker. Let's continue our discussion of NCERT Science series. And you already know we are discussing NCERT Science textbook for class 9 and we are doing a chapter by chapter analysis. In our previous interactions, we discussed about the concept of matter in the first two chapters. Then we discussed about the idea of atoms. And in our last interaction, I hope you remember the models defining structure of an atom. We discussed about Dalton's model, Thomson's model, Rutherford's model, Bohr's model, and Erwin Schrödinger's model as well. We discussed about atomic number and the mass number as well. Let's move on and discuss today chapter 5. Chapter 5, it is connected to biology. It talks about the fundamental unit of life, which is a cell. So in this interaction, we are going to understand the details about the fundamental and structural unit of our life. Our body is composed of billions of cells. So we are going to understand what are the constituents of the cell, what are the different cell organelles and their different functions. A very important discussion. Many different kinds of MCQs can be created from this topic. It is going to be the base of your biology knowledge. Moving forward, when we discuss about the 10th NCERT, 11th, 12th, we are focusing more and more on the ideas of biology itself. So without any delay, let us begin our interaction. But before that, if you're preparing for UPSC Civil Services examination, do visit our website www.studyiq.com because you will find the details of prelims to interview initiative there. This is the most comprehensive program for UPSC Civil Services preparation. If you have already enrolled for this, very good. I'll see you in the class. If you have not, or if you know anybody who is looking for UPSC Civil Services preparation, please spread the word because in this program, we will be hand-holding you during prelims, during mains, during interview. If you clear your prelims, then you will be called for a mains residential program, a kind of a Gurukul setup where you will be learning with the faculties, with your mentors. You will be focusing only on mains answer writing program. Mock interviews are also planned after that. So a complete package, just visit our website. The program is starting from 19 June. If you want heaviest discount on the program, do use my code Rahul Live. I'll see you there. Right. Let's begin our discussion of the fundamental unit of life. What that is a cell. We all know our body is composed of billions of cells, right? Our eye has different kinds of cells. Our skin has different kind of cell, right? The epidermis or the mesodermis, inside layers, there are different kinds of cells. Heart has different kinds of specific cell. So what is a cell? A cell is the fundamental and structural unit of life. In fact, when we talk about unicellular organisms, for a unicellular organism, this is life itself. A cell represents complete life. But for a multicellular organisms, cells slowly turn into tissues, tissues turn into organs, organs they set up themselves in some sort of a system. We have different organ systems because of which our body is running smoothly, our metabolism is running smoothly. So cell is the fundamental unit of life. And right from the beginning of, I would say, mid 19th century, 20th century, there has been extensive interest in finding out about this fundamental unit of life, that is cell. Scientists have been working on this. In our previous interactions, we discussed that cells are like the bricks, very similar to how we construct a building using bricks. Our body is made up of different cells, which are nothing but the bricks of this particular body. If you take out a simple onion peel and watch it under the microscope, you will find a structure like this, a very similar to brick structure, like a brick construction. As I told you, during the 19th century, 20th century, there was a lot of interest for finding out more about these cells. Initially, people started to view using the magnifying glasses, simple kind of microscopes. But by 20th century, we developed compound microscopes like this, where a small sample can be kept here and viewed from the eyepiece, and we saw beautiful structures of cells, and we were able to divide and distinguish among different cell organelles. If you look at the discovery of cell, it has seen a very interesting journey. Very earlier, during 1665 itself, Robert Hooke was the first person who found out or observed cells in a cork slice using a very primitive microscope. But for the first time, Anton Leeuwenhoek in 1674, he improved the microscope and discovered living cells in pond water. And for the first time, Robert Brown, he gave the idea of nucleus. In 1831, he observed different cells under the microscope and he said that every cell has a nucleus, which is like the central processing unit of that cell. The entire cell biology or the concept of cell was ultimately given by Schleiden and Schwann in 1830s. So many different kind of scientists have worked over this. Robert Hooke, Leeuwenhoek, there is Purkinje also. Purkinje in 1839, he coined the term protoplasm. Protoplasm for the fluid substance of the cell, that means cytoplasm plus the nucleus, he termed it as protoplasm. You must have heard about Purkinje fibers. We'll talk about that also in future. Why I'm discussing this? Many a time certain factual questions might appear about discovery of cell, discovery of the cell theory, cell biology, Schleiden and Schwann, and it was further expanded by Vichru. So today we have a great knowledge of different kinds of cells. Now, if we look at categorization or classification of cells, the functional and structural unit of life, we find two kinds of cells. One is a prokaryotic cell. We have discussed about that before also in our 6th, 7th, 8th interaction. Prokaryotic cell and eukaryotic cell. Prokaryotic cells are nothing but primitive cells. Now, whenever I mention about prokaryotic, I want all the UPSC civil services aspirants to remember it by bacteria, because we will be talking about bacteria. We will be talking about bacterial diseases, antibiotics, etc. in our future interaction also. So whenever there is a mention of bacteria, you need to remember that bacteria is a prokaryotic cell. And how do we differentiate between prokaryotic and eukaryotic? By understanding the development of its nucleus. Prokaryotic cells, they do not have a truly developed nucleus. In a eukaryotic cell, we do know that the nucleus is enclosed. If this is the cell, then this is the nucleus, and nucleus has an outer membrane, in fact a double membrane of the nucleus itself. But in prokaryotic cells, this is a classic idea of a bacteria, where you can see, let me just show you this picture, where yes, there is a cell membrane or a cell wall, which is the outer layer. There might be a cell wall also, but if you look at its genetic material, this genetic material is completely spread out through the cell. Whereas if you look at a eukaryotic cell, nucleus is somewhere here and the nuclear material is only enclosed within this particular part. Apart from this, prokaryotic cells, they lack cell organelles. That means there are no advanced cell organelles that are found in a prokaryotic cell. But do remember, prokaryotic cells, they do have ribosomes, special kind of ribosomes. We'll talk about ribosomes in some time. They have ribosomes for protein synthesis. In prokaryotes, you do find ribosomes, but not the other cell organelles. Apart from that, these prokaryotes are generally smaller. In the prokaryote, nucleus or a true nucleus is absent. In eukaryote, it is present. Both are, again, membrane bound organelles are missing. Outer membrane is present. Of course, the cell membrane is present in both. Please try to remember this. Cell membrane in this also, in this also. But the membrane bound organelles are missing. They are absent here. They are present on the other side. Cell structure is mostly unicellular. Apart from that, cell size is rather smaller, about 0.1 to 5 micrometers. The eukaryotic cell is a little larger, and eukaryotic cells are found in animals, plants, fungi, protists, and prokaryotic cells are nothing but bacteria. All right. Now, when we talk about this eukaryotic cell, as I told you, it is more advanced cell, where the nuclear material or the chromosomes or genetic information is enclosed within the nucleus. There are different or complex cell organelles which are present. That is why I told you. Let's take example of humans. I told you that there are billions of cells in our body. We can talk about a nerve cell, we can talk about a fat cell, we can talk about a blood cell, muscle cell, there is sperm, there is ovum. But overall, the structure of these cells can be different. But there would be a cell wall, there would be a nucleus, there would be specialized cell organelles in it. If we look at the cell structure, this is animal cell, this is a plant cell. In our previous interactions, we have discussed there are certain differences between the two and a lot of commonalities for sure. The differences are mainly connected to plastids. The differences are connected to cell wall. The difference is also connected to vacuole. We said plastids are only present in plant cells. Cell wall is present in the plant cell. Vacuole in these plant cells are large. Apart from that, when you look at the other cell organelles in both these, you will find a lot of commonality. You will see that both the cells will have nucleus, both the cells will have mitochondria, both the cells will have your endoplasmic reticulum, they will have your Golgi complex or Golgi apparatus, they will have plasma membrane, they will have ribosomes, they will have lysosomes, they will have mitochondria, and they will have cytoplasm. Yes. So let's talk about these cell organelles which are common to both these, and we'll also talk about some special ones here. That is the agenda for today's class itself. Let's begin. The first thing, cell membrane. Be it plant cell or animal cell, or even for that matter prokaryotic cells, they all have a cell membrane or a plasma membrane, which is the outer covering or the outer coating of the cell. Within this cell membrane or plasma membrane, different parts are there. For example, let's say this is nucleus. It is housed inside the cell membrane. This cell membrane is similar to the door of your house, right? I am just giving an analogy. Meaning, if something has to enter the cell or if something has to exit the cell, it has to pass through the door. It has to pass through the cell membrane. The cell membrane is made up of different proteins and lipids, and it is semi-permeable. That means it is going to allow things to go out. It is going to allow things to come inside the cell. Why? Because our body runs on metabolism. And for metabolism, what we need to do? We need to eat food. The energy has to go or enter this cell. Then whatever cell is going to metabolize, the waste materials are produced, and the waste material has to come out of the cell also. That means it has to be semi-permeable. And how does this help, or how does this occur exactly? The semi-permeable membrane, that is the cell membrane or a plasma membrane, it allows things to pass through it through diffusion or through osmosis. Now, for example, you all know I'm breathing right now. I'm breathing in the air. That means I'm utilizing oxygen. My lungs are now filled with oxygen, and this oxygen, whose concentration is now more in some lung cells, it has to be transferred to other cells. Oxygen, which is high concentration here, it is going to move into the cells where there is low concentration. That means the gas transfer, oxygen and carbon dioxide transfer, it occurs through the concept of diffusion. There can be osmosis also. Diffusion and osmosis are quite related. Diffusion, it occurs when there is no kind of semi-permeable membrane, and gas transfer, it occurs because of diffusion itself. And osmosis is a kind of specialized diffusion which occurs through a semi-permeable membrane, and here solvent transfer takes place. Solvent transfer takes place. Meaning, if my body wants water, when I consume water, the water has to enter the cell, and it has to pass through the cell membrane. It has to pass through the plasma membrane. How does it pass? It passes through osmosis. And that is why we see whenever a cell is put into different kinds of solution, it acts in a different way. For instance, if I put a cell in an isotonic solution means where the amount of solute and amount of solvent both are in some sort of equilibrium. For example, blood. If I put RBC in a blood cell, for that it becomes isotonic solution. Nothing is going to happen. That means the amount of water that is going out of cell and the amount of water that is coming into the cell, it will remain same. For plant cell, yes, it is going to remain flaccid because it will have more and more water in it. But as soon as I put the cell in a hypotonic solution, what is going to happen? Hypotonic solution means the outside environment has more solvent and less of solute. That means the solvent is going to come inside the cell. If it is a plant cell, the water is going to enter it. It is going to become more and more turgid. And if I put a cell in a hypertonic solution means where the solute is more, and in the environment solute is more, solvent is less. That means the water is now going to be lost from the cell, meaning the cell is going to lose water. This is called as plasmolysis. This is called as plasmolysis, and this process is basically called as ketolysis. Okay, just for your reference, it's called ketolysis, meaning the water is lost if I put a cell in a hypertonic solution, and the water is gained in a hypotonic solution. All right. Right. This is the job of your cell membrane or plasma membrane. Now, in plant cells specifically, I told you this is the cell membrane. I'll write it as CM. And outside the cell membrane, especially for plant cells, in fungi also, and in some bacteria, we find something called as cell wall. Now, the cell wall is mainly a means of protection. The cell wall, it is made up of different proteinaceous materials. It is made up of proteins and lipids, predominantly proteins itself. It protects the cell from mechanical damage and different kind of infections. It acts as a barrier. As I told you, if I put a cell in a hypotonic solution or in a hypertonic solution, it has to protect itself. How does it protect? It protects itself through the concept of cell wall. And please remember, cell wall is absent in the animal cell. It is present in plants, it is present in many fungi, it is present in many bacteria as well. Its main job is to protect the cell. You can see here the cell walls, it helps the cell to withstand very dilute hypotonic external media without bursting. If it is a very low hypotonic solution, there is a possibility the cell might burst. It will become turgid, more and more water, and eventually it might burst. Now, to protect itself, there is cell wall, and that is why you must have seen many of the bacteria, they can survive extreme conditions. Acidophiles, thermophiles, they can survive extreme conditions also, right? Cell wall. Cell wall also helps in regulating the osmosis. It maintains the osmotic pressure. Apart from this, every cell has something called as cytoplasm. Now, this is the cell membrane. This is the nucleus. This is the common drawing I'll be drawing it while explaining. Then there is cytoplasm, some jelly-like fluid inside the cell. This blue jelly-like fluid inside the cell, it is called as cytoplasm, right? Now, the job of cytoplasm is to house these specialized cell organelles and to maintain the fluidity. It is a jelly-like substance which consists of different salts, waters, and other organic molecules. It helps in survival of the cell. All right. So we discussed about cell membrane, cell wall, cytoplasm. Let's move on and talk about the central processing unit of the cell, which is nucleus. I told you, if this is cell membrane, the nucleus is enclosed. All right. The nucleus has double membranes. It has two membranes, and you will find genetic information in this, both in plants and animal cells. The nucleus is like the control center of any cell. Any activity by the cell, it is going to be controlled by the nucleus itself. The nucleus has a small nucleolus in it. It has two layers. It has two membranes, and that is the difference between eukaryote and prokaryote. In prokaryote, you will not find these membranes. There is a double membrane, and this membrane, it also allows movement of information, movement of material. This nucleolus contains your genetic information. The job of the nucleus is to house something called as chromosomes, or if the cell is not dividing, condensed form of chromosomes in form of chromatin or in form of thin wires is found. Whenever the cell is dividing, this chromatin, it divides. And these chromosomes, they are nothing but the information carriers of our genes. They are information of our inheritance. And protoplasm, the term protoplasm basically refers to the fluidic part of the cell. Cytoplasm plus the nucleus, it is called as protoplasm. Perkin give this name. Nucleus is the main center of the cell. For example, if there is any activity, if the cell has to grow, if the cell has to divide, the nucleus is going to send it information that it's a time for dividing. Favorable conditions have reached, and the cell is going to divide from 1 into 2. That information is found here. All right. Apart from this, cell also has specialized organelles called as endoplasmic reticulum. Now, This is the cell membrane, this is the nucleus. Very close to nucleus, we find different kinds of endoplasmic reticulum, which is like a communicating link between the nucleus and the other cell organelles. The nucleus has DNA. DNA communicates with the entire cell. How? DNA communicates through this endoplasmic reticulum. This sends information for protein production to ribosomes and other cell organelles. So endoplasmic reticulum is a large network of bound tubes. Let me just show you the image where you can just look at this particular picture of the animal cell, and you will see there are two kinds of endoplasmic reticulum: smooth endoplasmic reticulum and rough endoplasmic reticulum, just connected to nucleus, both of them. All right, right. So endoplasmic reticulum, its job is to ensure that it provides some sort of a skeletal framework to the cell. It provides stability to the cell. So it has different membrane bound tubes or sheets. There are two kinds of endoplasmic reticulum we find in a cell: smooth endoplasmic reticulum and rough endoplasmic reticulum. Take the pen here: smooth endoplasmic reticulum and rough endoplasmic reticulum. Now there is a difference among these two. Whenever I say endoplasmic reticulum, two kinds of ER can be found out. First one, smooth. Whenever I say smooth, there are no ribosomes present on this endoplasmic reticulum. But when I say rough endoplasmic reticulum, ribosomes are attached to it. Now, what are these ribosomes? See, these ribosomes are very important cell organelles in both plant and animal cells. It can also be found in prokaryotic cells, specialized ribosomes. Their job is to produce specific kind of proteins. And where do we get information for this? The DNA is going to send information to ribosome through the endoplasmic reticulum itself that this protein has to be produced. And you do know protein is extremely important for our body. Whenever I say human body is composed of water, yes. But when I talk about the next constituent, it is going to be protein itself. Protein is found in your muscles. Protein is found as chemicals. Your hormones are proteins. Protein is present in your blood, the globin protein. So protein is very important for us. Protein synthesis through ribosomes. And these ribosomes are attached on the rough endoplasmic reticulum. We can see them in the microscope, and these are, again, rough endoplasmic reticulums are the sites of manufacturing proteins. The smooth endoplasmic reticulum, it helps in production of fat molecules. It helps in production of fat molecules or lipids. Some of the proteins and lipids, they help in building of the different membranes that we saw: cell membrane, plasma membrane, etc. Some other proteins and lipids that are produced by endoplasmic reticulum, they also act as enzymes and hormones. And as I told you, endoplasmic reticulum, it helps in production and utilization of both proteins and lipids. It also helps in production of enzymes and hormones, which are needed for the cell for metabolism. Also, it serves as a channel for transport of materials. It also provides stability to the cell by giving it some sort of skeletal framework. Apart from endoplasmic reticulum, there is one more very important organelle called as Golgi complex or Golgi apparatus. You will see that endoplasmic reticulum is many a times connected to these Golgi apparatus. Golgi apparatus, they are a means of communication. If say endoplasmic reticulum is like a messaging unit from nucleus, it takes all the messages. Golgi apparatus or Golgi complex is a means of taking all this communication to other organelles of the cell. The name Golgi apparatus, it comes from a scientist, Camillo Golgi. Can you guess Camillo Golgi was from which country? Just take a guess and let me know in the comment section. Okay, so Golgi apparatus is again a bound organelle of eukaryotic cell. It consists of stacked pouches called cisternae, and these cisternae, they are basically means of transportation, modifying and packaging different proteins and lipids throughout the cell. They are a means of transportation and packaging. So Golgi apparatus, again located next to the endoplasmic reticulum, they are a means of communication. Yes, there are several kinds of Golgi apparatus. plant cell can contain hundreds of Golgi apparatus. In many cases, complex sugars are made simple in some Golgi complexes. Some Golgi apparatus and complexes are also useful in production of lysosomes. So what are these lysosomes? Lysosomes are basically the waste disposal system of a cell. Lysosomes are responsible for production of certain kinds of enzyme which help in waste disposal or digestion. Digestion or digestive enzymes are developed and they are capable of breaking down the organic material. Many a times when a cell itself has lost its utility, that cell also has to be dissolved, or the cell also has to burn down, and for that lysosome produces huge amount of enzymes so that that cell itself it digests, or it disintegrates, it decomposes. That is why many times lysosomes are called as the suicide bags of the cell. Apart from this, there is one more important organelle called as mitochondria. Mitochondria is called powerhouse of the cell. This is where the cellular respiration takes place. It is a place of cellular respiration. Now you all know that every day we have food, and when we have food we consume carbohydrates, proteins and lipids. These carbohydrates, proteins and lipids, how can they be utilized? They have to be turned into a common parameter which our body can use, and that common parameter or that common currency of our body is ATP, adenosine triphosphate, which is produced by the mitochondria. This mitochondria is going to use chemicals, right? Basic chemicals, that means the saccharides, the amino acids or the fatty acids that we consume, carbohydrates, fats, lipids. It uses oxygen in the reaction. Then it is going to produce adenosine triphosphate. Mitochondria, they have two membranes. The outer membrane is basically the membrane of mitochondrial organelle, but the inner membrane is highly folded. If you look at this inner membrane, it is highly folded. Why? Because the reaction has to take place. The Krebs cycle has to take place here, and ATP has to be produced. And for that, chemicals are utilized, oxygen is utilized, and ATP is produced here. So mitochondria, it produces the ATP, which is called as the energy currency of our body. And mitochondria, please remember this, it contains or it carries its own DNA information. You can see here, when I just look at this particular mitochondria, it has its own ribosomes. It also has its own DNA. It also has its own DNA. You can see mitochondria, it has its own DNA and ribosomes, right? In our future interactions, when we talk about biotechnology, we'll talk about mitochondrial diseases. We'll talk about issues connected to mitochondria. Please remember, mitochondria has its own ribosomes for protein synthesis. It has its own DNA information also. Apart from mitochondria, plastids are another important cell organelles specifically present in plant cells. Now, plastids are basically colored pigments or colored bodies in the plant cells which help them in photosynthesis or some other specific operation. One particular plastid, green color plastid, it is called as chloroplast. It contains chlorophyll. It contains huge amount of magnesium, which helps in photosynthesis in plant. That is why plants are called autotrophs, because they can undergo photosynthesis, they can produce their own foods. But please remember, plants do not contain only one kind of plastid called as chloroplast. They can also contain different kinds of plastids like leucoplasts, like amyloplasts, etc. From a perspective of plant body or plant cell, these plastids or chlorophyll, it can be considered similar to mitochondria because they are the areas of photosynthesis. They produce food. All right, plastids specific to plant cells. Apart from this, there is an organelle called vacuole also, and in plant cell, if this is the plant cell, vacuole is generally very big. These vacuoles are storage spaces. These vacuoles are storage spaces. Why do they use? You can see the root word here also, vac. The root word is vacuum. These are storage spaces. What do they store? Please remember, vacuoles, can store anything. Vacuoles can store gas, vacuoles can store water, vacuoles can store nutrients, vacuoles can store even waste material. Very important pointer here. Vacuoles in the plant cells are bigger in size because the requirement of plant is more. It can store water, it can help in gas transfer, CO2 and oxygen transfer for plant. It can also store waste material. In plant cells, the vacuoles occupy about 50 to 90% by volume itself. Clear? Right. So there are vacuoles also. Apart from this, whenever we talk about cells, we do know that cells divide. Now you can see that I met with a small accident and there is a small cut here on which I have applied the band-aid. Now what is going to happen? Today you might see a small amount of scar or small amount of injury there, but maybe after 8 days, maybe 10 days, when I remove this band-aid, you will not see anything. I would be healed. Why does this happen? Because cell undergoes division, and the eukaryotic cell, it undergoes two kinds of division. One is the normal mitosis. Now this applies to all the eukaryotic cells which are not gametes. Please remember, mitosis, it applies to all the somatic cells. What is this mitosis? Mitosis is a process of cell division where you will find that the cell undergoes different kinds of phases. We'll talk about these phases in our subsequent interactions. But when I talk about mitosis, the first phase is called as interphase, where the chromatin or chromosome is duplicated. All the chromosomes are duplicated. Then it undergoes 4 different phases: PMAT, that is prophase, metaphase, anaphase, and telophase. Undergoing these phases, ultimately one cell, it divides into two daughter cells, and it is because of this it will be healed. So mitosis is a division of somatic cells which is responsible for our growth, which is responsible for healing. The significant contribution of mitosis is cell repair. Our body constantly loses cells, our body constantly needs cells, right? And for that, the cell has to undergo mitotic division. Mitosis leads to division of a cell into 2 diploid daughter cells, meaning there is no change in the chromosomal information. Please note here that the cell where it has started, imagine if I talk about human cell, it has 23 pairs of chromosomes, correct? It has 23 pairs of chromosomes. That means totally 46 chromosomes. In mitosis, when this cell divides and 2 different cells are created, meaning these 2 cells will have 23 pairs of chromosomes. They are going to have completely 46 chromosomes. It's not half. That is why it's a diploid daughter cell. 2 diploid daughter cells are produced. There is another kind of cell division in cells, in sexual animals, or especially with the gametes. It's called as meiosis. Meiosis is a specialized kind of cell division where a cell divides into haploid daughter cells where the chromosomes are halved. In organisms which undergo sexual reproduction, you know, a gamete or a set of chromosome comes from one parent, another set of chromosome comes from another parent. The two are going to fuse, form zygote. Then the organism which is born, the offspring, it is going to have complete set of chromosomes. It happens through the process of meiosis. Now meiosis takes place through 2 kinds of division where a single diploid cell, that means where you have 46 chromosomes, this particular cell, it divides into 4 cells which are haploid. 4 cells which are haploid. That means these 4 cells are going to have 23 chromosomes each, half the chromosomes than the parent cell. And this is responsible mainly for inheritance, meaning in our body there should be constant genetic information or inheritance based information. Half set of chromosomes come from father side, half set of chromosomes come from the mother side, and an offspring or then the next generation individual, they will have complete set of chromosomes, half from one side, half from the other side. Now this happens because of meiosis. Now please remember that mitosis and meiosis, mitosis occurs in somatic cell, where DNA replication takes place in interphase. We'll talk about these phases in our subsequent discussion, subsequent classes, right? Meiosis, it takes place through two kinds of division, where ultimately the result of daughter cells that are produced. For mitosis, it produces two diploid cells. For meiosis, it produces four haploid cells. Let me explain it clearly again. In mitosis, please remember, in mitosis, you will see that complete set of chromosomes can be seen here in the two daughter cells, complete set of chromosomes. But when you talk about meiosis, you will see that four haploid cells are produced where only half the chromosomes are transferred. These half chromosomes will join with the other half coming from the other counterpart, that is half from father, half from mother. Please remember this cell division process, because this is the base of our biology knowledge, and we are going to understand more and more about these things in our subsequent discussions, because biology is very important for us. A lot of questions do appear from this. All right, so this was about the fundamental unit of life, that is the cell organelles and cell division, mitosis and meiosis. Thank you for watching this. And if you have liked this video, you can always follow me on this particular handle at the rate Rahul Sai 222. Thank you again. Jai Hind.